Hello students, in paper Molecular Biology and Genetic Engineering, today's topic for discussion is blotting technique. Then first question comes in your mind that what is blotting? Here blotting is nothing but the method for detection of specific biomolecules in a sample of complex position, because whenever we are having a sample, it may be a mixture of DNA, RNA and the proteins, or apart from that any components is there. So we have to detect with the help of this blotting technique, and this is very much near to the electrophoresis, where we are using this method for separation of the DNA, RNA and the protein, where they are separated on the basis of their size as well as the charge. Afterwards they are transferred to the nitrocellulose membrane, and this is followed by the hybridization with the probe. Then what are probes? These are the strands of either DNA or RNA or the proteins. Strands in case of a DNA and the RNA. In case of a proteins, they are either antibodies which are having similarity with the present complex solution, and they are having DNA or the RNA, or it may be the proteins. Afterwards there is the hybridization, which is with the help of radioactive probes, and most of the times P32 is used. This is because it is having high energy and easy to incorporate into the phosphate group of the DNA TPs, and it can be easily detected by the X-ray film. There are major 4 types of a blotting technique depending upon what type of a molecule we are going to detect. So this southern blotting is for detection of DNA, then northern blotting is for the RNA, western blotting is to detect the protein or the antibodies, and dot blot technique. Here the different form from other 3 techniques, we can analyze or we can detect DNA, RNA and the protein, but we are using the sample in the form of a dot. That's why this is a dot blot technique. We'll go first for the southern blotting technique. This term is a southern blotting because the name of its inventor, and he is E.M. Southern, where DNA hybridization that forms its basis. This is because the name is given southern blotting technique. The first step for this blotting technique is we have to cleave the DNA with the help of a restriction enzyme. Whatever compound or whatever complex mixture we are having, first we have to cleave that DNA with the help of a restriction enzyme, and it is followed by the agarose gel electrophoresis, where the DNA molecule is separated on the basis of their size. And basic idea behind the electrophoresis: so DNA is negatively charged and it moves toward the positive charge, and with the help of this we will get different bands of the DNA depending upon their size. So smaller size DNA molecule, they can run fast, and obviously the larger size DNA molecules, they remains or they lag behind. Then afterwards this gel is transferred on a nitrocellulose filter. This second figure, you can observe how this nitrocellulose paper, and below which there is agarose gel, again there is a sponge and alkali solution, and above the nitrocellulose paper there is a use of a stack of a paper towel, so that the bands from the DNA which are present on the nitrocellulose membrane, they can be transferred on the on the agarose gel. They are transferred on the nitrocellulose paper. Afterwards we have to remove the nitrocellulose paper with tightly bound nucleic acid. Then this nitrocellulose paper is hybridized with radio labeled probe, and many of the times P32 is used, that already we have discussed in the first slide. So in the bag, which is lock and seal bag, where the nitrocellulose paper and the solution which is with the probe is kept for the hybridization. What is probe? So this probe, it is with the again DNA segment, and it can form a complementary strand where it shows the similarity with the agarose gel. So afterwards, this labeled probe hybridizes to the complementary DNA strand. How this can be visualized? By the autoradiography. In autoradiography, we will get finally the bands which are similar in our sample. They can form a complementary strand with the probes which are present in the radio labeled probe. So dark bands at the end shows position of a DNA sequences that hybridizes with the probe. So this is all about the Southern blotting technique, which is useful for detection of a DNA fragment in a solution. Now let's go for the importance or the applications of Southern blotting. It is useful for gene discovery as well as the mapping, evolution developmental studies, diagnostic as well as the forensic studies. Here we can determine the molecular weight of a restriction fragment with the help of electrophoresis. It also gives us idea about the particular bit of a DNA in the sample, and we can identify the different mutations, deletion, insertion as well as the gene rearrangement. This is also useful for prognosis of a cancer and prenatal diagnosis of a genetic disease. One of the disease, that is leukemia, it can be also identified with the help of this Southern blotting. Also HIV and other infectious diseases can be identified with the help of this technique. So this is all about the Southern blotting technique, which is useful for detection of the DNA fragments. Then another technique is for the detection of the RNA fragment in the complex mixture. So the steps which are similar to the Southern blotting, firstly we have to run the RNA segment through the agarose gel electrophoresis. Why the agarose gel electrophoresis is used? Because this agarose as well as the polyacrylamide, that is useful for separation of the DNA fragment as well as the RNA fragment depending upon their size. If the DNA fragments are having more size, they can be separated agarose gel with pulsed electrical field or the field inversion. But interestingly, in Northern blotting, there is no use of a restriction enzyme. Because why this is so? Because RNA is a single stranded and it can fold into the various conformation, and this can be achieved through the intramolecular base pairing. So through the electrophoresis, separation of the DNA fragment depending upon their size is carried out. Then as per the Southern blotting, it is transferred to the nitrocellulose membrane with the help of diisobenzoxyl methane paper. Why this is used? Because as compared to the nitrocellulose membrane, this paper can be used for the several time, and it shows its peculiarity. Apart from use for the several time, it remains with its character as it is when we are using for the several times. Then after this blotting, we will get the RNA fragment from this agarose gel to the nitrocellulose membrane. Again, this is hybridized with the probe, and finally by autoradiography we will get the separation of this DNA fragment when this DNA probe is hybridized to an RNA transcript. So this technique is useful for detection of RNA in the complex mixture. This Northen blotting technique is useful for a gene expression pattern, either in the tissues or at different developmental stages, and along with this environmental stress levels, pathogen infection. So expression of the gene at this different responses can be achieved or can be identified with the help of Northen blotting technique. It is also useful for overexpression of oncogene that cause the cancer. Then downregulation of a tumor suppression genes in cancerous cell when it is compared to the normal tissue. It is also achieved through this blotting technique. Apart from this, rejection of a transplanted organs, how the gene are expressed in this organ, this is also useful for the Northen blotting technique. This is in short for the RNA fragment detection and its uses. Third technique is for detection of a protein, and that is Western blotting, or we can say protein blotting, or this is also known as the immunoblotting. And here we are using the same steps, but instead of agarose we are using polyacrylamide gel electrophoresis with SDS detergent. Then this gel is treated with the Kumasi Brilliant Blue, and afterwards this gel is transferred on the nitrocellulose membrane. The same idea is here, blotting. Then here you can observe in the figure how the stack of the paper towels, then nitrocellulose membrane, and the gel which is with the protein fragment is over here. Then immobilization of proteins in a solution of primary antibodies. Then in this step only primary antibodies are detected and they can be founding bond. Afterwards washing of non-bound antibodies, and afterwards this solution is transferred into the secondary antibodies containing solution where all antibodies can be detected. Finally it is placed or it is subjected to the autoradiography and we will get separation of proteins. Here detection of antibody by other antibodies with some fluorescent or color production marker system is carried out. The idea behind the protein blotting is antibody binds to the antigenic site of a target protein, and that's why the name is protein blotting or the immunoblotting is given. This is useful for disease diagnosis. Along with this, size as well as the amount of protein can be determined. HIV confirmation test is carried out with the help of Western blotting technique by using patient's serum. Along with this, defective proteins can be detected, like this separation of the proteins, and definitive test for the Lyme disease, hepatitis as well as the herpes, they are also can be identified with the help of Western blotting technique. Here this figure gives you idea about how this enzyme converts substrate to give us visible colored product, and that can be visualized in the membrane. So this target protein, it binds with the primary antibody, then secondary antibody, then enzyme conjugate, then it is finally the substrate product, and they are emitting the specific light, and this orange colored part you can observe and it can be visualized in the membrane. The last technique is dot blot technique, and it is different from previous technique because we are using non-electrophoresis sample. Either it is DNA or the RNA or the protein which is without non-electrophoresis is used for this dot blot technique, and the sample is in the form of a dot. Finally, in this dot blot technique, a well is there, and we can at a time number of samples can be analyzed with the help of this dot blot technique. We can apply directly the small dots of either DNA, RNA or the proteins on the nitrocellulose membrane, and afterwards denaturation of this DNA, RNA or the proteins and immobilization by baking at the 70 to 80 degrees Celsius for 2 to 3 hours. Then we have to add the labeled probe for the hybridization and incubation of probe that binds to the complementary strands in case of DNA and the RNA, and in case of a protein, antibodies. Finally, washing of excess of unbound probes, and this last figure gives you idea about after autoradiography black dots. These black dot indicates presence of sequences in that sample, and this is with the help of a probe, because probe has the similarity with the either DNA sequence or protein or the protein or the RNA or the protein samples in our complex mixture. Here, interestingly, intensity of dot is relative to the concentration of sequences related to the probe. So with the help of how intense the dot is there, amount of the sample or the amount of DNA, RNA or the protein can be identified. But one drawback is there for the dot blot technique. We will not get the size of that DNA as well as the RNA and the protein molecule because there is no electrophoresis. But this dot blot technique detects biomolecule in a single run. Lot of samples can be analyzed in a single run. Along with this, specific sequence of gene can be detected in a transgenic individual, and it confirms presence or absence of a biomolecule which can be detected by DNA probes or the antibody in case of a protein. And this is useful for screening viral genomic DNA RNA transcript. So this is all about the different techniques. This last figure gives us the comparative difference between the southern blotting technique, which is for the DNA detection, northern for the RNA detection, and western blotting technique for the detection of protein. Use of agarose gel electrophoresis, and for protein polyacrylamide gel electrophoresis is carried out. And probe membrane single stranded complementary DNA or RNA to specific sequence with the help of this technique. And finally, we'll get the similar sequences in our sample, those forming the complementary strand with the probe in case of DNA and the RNA, and in case of the protein, antigen and antibody. So here in western blotting, the sample contains specific polypeptides can be identified. In northern blotting, sample containing specific RNA transcript can be identified, and in southern blotting, specific sample containing DNA restriction fragments. And the fourth technique we have studied, that is dot blot, where use of a dot in the use of a sample in the form of dot can be achieved. So with this, I am going to stop here. Thank you.